When we look at the composition of the human body, we see a remarkable balance between solid structures and fluids. As shown in this pie chart, about 40% of your body mass is made up of solids. So this includes bone, muscle, protein, fats, and other structural components. The remaining 60%, which is very similar to how much seawater is on the world in general. But the remaining 60% is fluid. And that fluid gets divided into three main compartments. The largest share, 33%, is found inside of our cells, and that's the intracellular fluid, or the ICF. This fluid is essential for cellular environment. It supports chemical reactions in cells and it helps cells maintain their structure. Interstitial fluid, IF, is what bathes and cushions our cells, and it makes up about 22% of your body. And then there is plasma, which is the liquid portion of blood. And that accounts for the smallest portion of liquids in your body at 5%. These fluid compartments, they're carefully regulated to ensure that our cells stay hydrated and that nutrients, waste, and signals can be moved efficiently throughout the body. This bar graph is a graph that highlights the distinct electrolyte makeup of the three fluid compartments, the ICF, the plasma, and the interstitial fluid, IF. Starting with the ICF, the dominant ion is potassium, seen here at high concentrations of 150 milligrams per deciliter. This makes sense because potassium is essential for generating resting membrane potentials, and it's actively transported into cells by the sodium potassium pump. Also notable in the ICF is the high level of phosphate and proteins, both of which serve as important intracellular buffers and structural components. In contrast, both the plasma and the interstitial fluid, which make up together the extracellular fluid, both of them have high concentrations of sodium and chloride. Plasma has about 125 milligrams per deciliter of sodium, while interstitial fluid is slightly higher than that at 150. These ions, they help maintain fluid balance and nerve signal transmission. Plasma also contains more proteins than interstitial fluid, mainly albumin, right, which helps create that oncotic pressure or that osmolarity to pull water back into the bloodstream. You can also see the bicarbonate levels are similar in plasma and interstitial fluids, and that kind of reflects their shared role in buffering blood pH. This compartmentalization of electrolytes, it's really needed for cell function. The selective permeability of membranes, like what leaks in and out, and the active transport mechanisms like the sodium potassium gradient, the sodium potassium pump, all of those things, they maintain those gradients that allow action potentials, right? Those gradients allow you to have thoughts that control different areas in your body. Recall from our very first topic that there are fluid compartments in the body. The intracellular fluid, or the ICF, is all the fluid contained in your cells. It's as if I took each cell and poured out all of the contents into a glass. That is your ICF, and it makes up about 40% of the fluids in your body. Your ECF, or extracellular fluid, is all of the other fluid, right? But this compartment, the ECF, it has its own two subcompartments. Extracellular fluid can be found in the plasma portion of whole blood, and that makes up about 4% of your fluids. That would be found inside the blood vessels. But extracellular fluid also contains the interstitial fluids, making up about 16% of the fluids in your body. These are the fluids that your cells are bathing in. Your plasma exchanges with the outside world by way of your digestive system, respiratory system, urinary system, but then the plasma exchanges with your interstitial fluids. And then these interstitial fluids, they then exchange with the cells themselves, the intracellular fluid. So there are two fluid compartments. The things like oxygen, carbon dioxide, sodium, potassium, glucose. There are two compartments that they have to go through to get to the cells. And gradients have to be set up across these compartments for things to move down their gradients. This compartment of interstitial fluids is very important to this chapter, right? That interstitial fluid lies between the blood that wants to give you all of these great things like oxygen and glucose, and it stands between the blood and the intracellular fluids, right? So it's this interstitial fluid, this compartment that bathes our cells, now becomes extraordinarily important because it is the fluid compartment that exchanges with blood and with our cells. When your body manages osmolarity, which sodium is the primary contributor to, it uses fluid shifts. It doesn't do this by adding or deleting electrolytes or solids or even water. It does this by moving around water. That's what a fluid shift is. It's a rapid movement of water between compartments. So a compartment could be the plasma or the ICF, or the ICF or the IF. So if you drink a lot of water, you're going to flood your plasma with that water. And a lot of it is just going to shift to your interstitial fluids. Officially, this is your body adjusting the volume in an effort to control osmolarity as the major set point. So you have to remember that in order to control many of these solids, we just adjust the volume. And you can do that by adding water via thirst or peeing water by way of dilute urine. In my general biology class, we talk about ions, but here in anatomy and physiology, we extend that a little bit and talk about them as electrolytes. It's important to realize that you have electricity running through your body, and the current has a very easy time doing that because you are a water-based organism. So a thought is really just the movement of electricity. If you move your big toe, electricity just zaps through your spine, down the sciatic nerve to bring the message to your toe muscles that they should contract. And that electricity that your body uses to move around information, it depends on the ions that are present in your blood. It depends on what is called your interstitial fluids, right? The fluids between your cells and your cells themselves. Therefore, we call all salts electrolytes, which is a word that denotes the ability to conduct an electrical current. So I guess you could say all ions are electrolytes, but not all electrolytes are ions. When a salt is absorbed into your blood, which is a water-based substance from your small intestine, it immediately breaks apart into its corresponding anion and cation. So the salt you ingested at lunch immediately split into the sodium cation and the chloride anion when it hit your blood. This splitting is called dissociation. That is what you see in the flask down here on the bottom. The cations and anions are fully
separated with that strong electrolyte. Cations and anions in a weak electrolyte also separate, but not all of them. So as you can see in the picture, weak electrolytes don't dissociate completely in your blood. Anything that isn't an electrolyte doesn't dissociate. The molecule in the flask on the left, it could be an alcohol or something, which stays fully intact when you put it into water. It is a non-electrolyte. It does not contribute to the conduction of that electric current.
Officially, osmolarity refers to the number of osmoles of solute per kilogram of solvent. Yeah, that means very little to me. So it measures how many particles of solute are present in a given mass of solvent. Imagine you're preparing fruit punch and you are interested in how many fruit pieces, and that's your solute, are present in each liter of the punch. That's your solution. And can I just say, this sounds more like sangria than fruit punch? You are focusing on how many fruit pieces are present per kilogram of just the juice, and you're ignoring the volume added by the fruit itself. In clinical practice, osmolarity is often preferred because it's measured based on the mass of the solvent, and that makes it less susceptible to changes in temperature and pressure. I think it was Charles's law, right? Says that things expand with temperature. But osmolarity provides a more accurate assessment of a solution's concentration, especially when there are physiological conditions happening. Osmolarity, being volume based, can be influenced by environmental factors that affect volume, such as temperature fluctuations. But in general, if your osmolarity is high, you are dehydrated despite how much blood is in your body. If your osmolarity is low, you are well watered despite how much volume of blood is in your body.